Good morning. If we think about this course where we are, we're in a part of a course now that deals with specificity, the ability of biological systems to work precisely, despite the problem that for every chemical reaction that's designed, that's right, nice deep sigh of relief, there's many other chemicals inside the cell that look just like the chemical we're trying to identify. And that's a big background that we want to minimize the effect of this chemical background. So that's what's called specificity. So if we think about our course, if we had one part about network motifs, the idea that complicated networks are made just of a handful of small recurring circuits, each one with specific functions. And then we talked about robustness, which is circuitry, how circuitry is designed so that its dynamics is invariant to variations in the concentrations of the proteins that make up the circuit. Yeah? And then we talked about evolution and evolution and optimality. How natural selection can come up with all these mechanisms. And now, in this part about specificity, which means what we're trying to look for are general, universal kind of mechanisms for reducing the errors caused by misreading the target. So again, the idea is that we can look for, if we look for, we'll find mechanisms that can unite different systems, very different systems. Different systems in their details, but they're united by a general principle. Like last time we saw kinetic proofreading and conformational proofreading, which use a kind of seemingly wasteful step in order to achieve specificity. Kinetic proofreading has this energy, and a step that costs energy and kind of effectively causes a delay. And during this delay, some of the correct signal falls off, but the incorrect signal falls off much more. And we're left with less correct signal, but much higher ratio of correct to incorrect. There's a higher specificity. Or conformational proofreading, where we pay this deformation-free energy, and we pay to bend the protein, to hug, and they try to hug, right? And because you pay this deformation-free energy, the wrong target just can't pay it. It doesn't have enough hydrogen bonds to stay bound. But the correct target does. And if you tune it right, so the deformation energy is like in the middle between the two binding energies, you can have a very large increase in your specificity. So again, these seemingly wasteful details always pick up my attention when I see something that doesn't have, it's not the most minimal mechanism, or has something wasteful or paradoxical or elaborate. I wake up and I say, oh, maybe there's something to understand here. That's general. Okay, so we'll continue in this spirit here. We talked about kinetic proofreading in this lecture, and today we'll talk about demand rules for gene regulation. It has to do with specificity, and again, a general principle. It's kind of simple and nice to teach. So that's the plan for today. So the question of rules, I want to tell you the history about gene regulation. This idea that there's DNA, right? And genes are DNA wasn't clear until around, some experiments in the 40s until around the 50s that this stuff has genes in it. And the idea that proteins can touch the DNA and turn on genes was nobody came up with this idea. It came through experiments by Jacob and Monod on the lac system, showing that the proteins that eat lactose, lac-Z, isn't made all the time. It's only made when there's lactose around and no glucose. So like a two-input decision. And they worked hard to figure out how that happens and found that there's this lac repressor that binds the DNA, and it falls off when there's lactose, causing expression. There's another protein called CRP that binds there to activate if there's no glucose. So there's two regulators. This whole idea of regulation arose there. And at first, Jacques Monod found a repressor in the lac system, and they were working on another system, a virus that infects bacteria, and they also found a repressor there. So in two different systems, they found the same principle, repressor that binds DNA, and they were thinking there's only repressors. Biology has only repressors. Nice deep sigh of relief. Hi. I recognize you guys already, you know, so it's nice to see you. But then, you know, and it's very elegant because with repressors you can do you can get a positive, you can have a repressor of a repressor, which is a positive regulation, right? So you can do anything with minus signs. You can get pluses by minus. And it was very pretty. But then there was this bothersome scientist that said, no, there's also activators. Activators are different from repressors. When they bind the DNA, they activate the gene. And when they go off the DNA, they turn off the gene, right? Repressors, when they bind, they turn off the gene. So. And the dogma was so strong that it was very difficult for them to publish their paper. But they did. And it turns out in the Arabino's system, it turns out that there are also activators and repressors. Okay, so there's two kinds. And then more and more genes were figured out. And some genes had activators, some genes had repressors. Each kind of repression has its own mechanism, etc. And the hope that there will be just one kind of control was vanished. And the same thing with all kinds of biological mechanisms. Each gene, each protein, etc., had its own kind of intricate mechanism. And the idea of searching for simple rules became kind of unpopular. In the 80s and the 90s, and people were running gels all the time and discovering, hi, welcome. Discovering these very important proteins to do, you know, what biology. But the idea that there's general rules was very not something people were looking for generally. Of course, there's exceptions, like Michael Savageau. Other people were looking for rules all the time, even in that period. It just seemed like a zoo of mechanisms, right? But now we're in the 21st century, and and it turns out that that all this richness of mechanism, you can, as you see in this course, form rules, principles to understand them. And but maybe the first one, or was by Michael Savageau, asking this very simple question: why are some genes regulated by activators and other genes by repressors? So that's the question we'll ask. I'm going to spell it out. I would say it's even a philosophical question, whether when you look at biology, what do you look for? Is it the differences between systems, or the uniting principles between systems? And of course, you need to do both. But there's certainly a disproportionate amount of splitting in the last decades in biology. That's something we go against that in this course, just to correct things. So in Savageau, he was trained in literature, by the way, and then in electrical engineering. Michael Savageau, 1970s. Why are some genes regulated by activators and others by repressors? So the idea that it's important to realize, okay, that you can get a gene turned on by a signal by either mode. Mode, when I say mode, I mean activator or repressor. Just very simply, okay, you can turn on a gene by having a repressor go off the DNA or by having an activator go on the DNA. You can turn on a gene, hi, nice deep sigh of relief. Welcome. Let's start with a repressor. So you have the repressor, and you have your signal, and they make it this complex. And it's the repressor that binds the DNA, right? So the repressor binds the DNA, and there's no transcription. What I'm trying to say is that when the repressor binds the DNA, it's off. And when the repressor is off the DNA, the gene is made. It's on. Right? And that happens when the signal binds to it. For example. So you add the signal, you get the gene expressed. An activator when it's bound to the DNA, the gene is on. And when it's off the DNA, the gene is off. So let's say it can bind the DNA with when it's connected to the signal. Right? So, so at first glance, there's no difference. There's no fundamental difference. I mean, and you might think. You might think that perhaps choice of mode, activator, repressor, is random or historical precedent, whatever happened to evolve first, right? That's certainly a hypothesis. But we don't want to resort to this hypothesis unless we have to. That's the philosophy, okay? So just think, what could be the difference? Now, Savageau looked at this problem and he noticed a correlation. And his correlation is that are called Savageau demand rules. And it says that genes needed often in the natural environment which he called high demand genes. So genes that often, a large fraction of the time, need to be turned on tend to be regulated by activators. Low demand genes, that is to say, genes that are regularly turned off but rarely turned on by repressors. I don't want you to get confused by how strong the gene is turned on. There's not a statement of genes that are turned on strong, there's say 100,000 copies of the protein, or weak, 10 copies of the protein. That's not the point. The point is, when the gene is on to its maximal level, is that a large fraction of the time in the natural environment, or a small fraction of the time? It's not weak or strong genes, it's rarely needed or commonly needed genes. That's called Savageau demand rule. And it's the evidence for it is in E. coli. So let's restrict it right now to E. coli. We can talk about other organisms later. Okay. So despite the equivalence of the two mechanisms, like there's some difference between the genes that are correlated by that are regulated by activators and regulated by repressors. Any questions so far?
Isn't it a little bit counterintuitive in kind of energy conversion kind of aspect? Because intuitively, I'd say if something is high demand, then a repressor would be more efficient because the default would be that the gene is active all the time. And then when in like the specific times when it's not needed, then we'll have the repressor repress its expression, while like the default for low demand would be that it's shut off, and only an activator will activate.
So you're saying it's more economical to have an activator for a low demand gene. Why would it be more economical? I just want to remind you that these activators and repressors are around, and you don't need to make them. Let's assume they're around all the time. Or in other words, you need to produce them all the time. The question whether they bind the DNA or don't bind the DNA doesn't really cost the cell energy. So is that still relevant to your thinking? If you had to make the activator. Yeah.
But their production does, and I'm assuming like the repressor needs to be produced as long as the gene is not needed, right?
Right. The regulators, let's think about it. I mean, there's of course exceptions. The regulators in general are produced all the time at not very high levels, and their function is to sense the signal and then bind or unbind to the DNA. So they need to be there all the time. So otherwise, this kind of economy is exactly a kind of a theory that we can use to separate the two. But let's assume now that they're both produced. That's important. Okay, so you don't separate them. Yeah.
But following that reasoning, if they're there all the time, then their levels must be different.
Why?
It's regulated by a repressor. Then the repressor must be expressed at high levels.
Why is that?
In order to shut down everything. And then the repressor needs to be down-regulated in order for the gene to be expressed.
Okay. So the comment is that a repressor needs to be at higher levels than an activator, higher protein levels than an activator.
Assuming basal state.
Oh, in order to avoid a leakiness.
Yes.
Leakiness. Good. I mean, that might be something general. I mean that repressors, in order to avoid leakiness, you need to make more of them. If you want non-leaky, and activators, you want
proportional to expression level.
Proportional to expression level. So that's like a fundamental difference between activators and repressors, right? And then I'd say try to think if that corresponds to this then, because you might say for all genes I want to have either no leakiness or yes, a little bit of leakiness. Some genes do. So see if you can think if it can correlate to the demand. These questions go to, I think from these questions, both of your questions, you can see that it's very actually difficult to say the two mechanisms are exactly the same. And that's one of the points, is to assume the two mechanisms are completely equivalent is rare. Example for the demand rules, the lac genes that we're familiar with are repressed by the repressor lac I. The signal is lactose. Now, in order to understand demand, it's actually quite It's quite difficult, because what you need to know is look in the natural environment of E. coli, where it lives, and understand what's rare and what's not rare. It's not in the lab. It's in the, where does E. coli live? It lives in our gut, in our intestinal system, and then it goes out and then goes back in different ways. So we have to think about this sugar lactose. Is it rare or not rare? And lactose, it's nice, because you can probably make a convincing case. Where does lactose come from? It comes from mother's milk in mammals, and that's it. So you have E. coli in cows or in mammals. So that's a very small fraction of time. And in human beings, it comes when we eat cheese and milk and stuff like that. And lactose, as you remember, is glucose connected to galactose. And our body has these, our own lactase genes, and it cuts up lactose very quickly up in our intestinal system. And where E. coli lives, which is mainly down there in the colon, it doesn't see the lactose. So it does see the galactose only rarely when it passes through the upper intestine. And then it gets cut up into galactose and glucose, so it sees lactose together with galactose and glucose for maybe 1 generation out of the 3 days that it spends in our body. An hour out of the 3 days that it spends in our body. So lactose is a rare sugar in our gut. Therefore, the lac system is low demand. Therefore, it's regulated by a repressor. That's a kind of evidence. Okay? Did I explain myself? On the other hand, if you look at the colon down there, what sugars do make it down there are the sugars where we don't have natural pumps in our intestine to drink them up from the food. Also, sugars that's found in these dietary fibers, all these plant fibers. Plant fibers are polymers a lot of times of sugars. Xylose, arabinose, xylose. So those sugars, those plant fibers are broken down by bacteria in our colon, and those sugars appear there. So an example of that is the sugar, another sugar we talked about, arabinose, is common in the gut. Therefore, the ARA system that we talked about a little bit, this system is in charge of transporting and breaking down arabinose for use as a carbon and energy source, is in high demand in the natural environment of E. coli, and it's regulated by an activator. And indeed, it's regulated by an activator, RC. This was actually the first activator, the one that couldn't get published for 10 years, this RC. Okay? And all right, so those, all right, and then Savageau went on to catalog, according to the evidence that was available back then. And for you guys, is anybody here working on microbiota, like gut microbiota? So, so today the idea of what's the environment of bacteria in the gut is a very hot topic. And I bet there's much more better data today about what sugars are down there and how they change from person to person. But if you try to write down some sugars like this, I'm going to write down some sugars, okay? Arabinose, these names, arabinose, fucose, sugars, yeah? Galactose, glycerol, lactose, maltose, okay? And then see in the gut what's going on. You see high, high, low, 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 high, and then you ask what the mode is, and you have the positive mode, arrow C here, positive mode, FUCR, the low mode, GAL-R, repressor, I mean, glycerol has a repressor, GLP-R, lactose has a repressor, LAC-I, and maltose has an activator called MAL-T, right? Rhamnose is high and has an activator RAS. Okay, so evidence like this for 100 different metabolites or gene systems came out basically consistent. Now I want to say it's hard to get accurate demands. Maybe now one of you guys can do that. I mean, get what's actually going on in the gut. But let's take a nice deep sigh of relief. All right, so I just want to open your mind that we know much more about the genes of E. coli and the circuits, much more than we do about the natural environment in which it evolved. So one kind of project you can think of is kind of, you might call it reverse ecology. Finding the natural environment from the molecular design. Okay. Similar, you can think about degradation systems. So if you have a lot of amino acid arginine outside, then E. coli does. Welcome. Nice deep sigh of relief. There's a system inside that makes the amino acid arginine. If there's a lot of it outside, that system of making arginine inside the cell is in low demand because you can just transport arginine from the outside. So you can make the same arguments for biosynthesis systems. And then you can also have evidence, like without even knowing what the natural demand is, if you have two systems with opposite functions, like transporting arginine from the outside or making it from fresh from the inside, they have opposite functions, you can expect them to have opposite demands. And there you expect them to have opposite modes of regulation. Let's just say, the biosynthesis system, if it's regulated by an activator, the transport system of the same thing from the outside will be regulated by a repressor. And you see that. And there you don't even need to know the demand. So if you have two systems that do the opposite things, the demand rules predict that they'll be regulated by opposite modes. For the arginine case, indeed, the arginine transport is regulated by an activator, and the arginine biosynthesis regulated by a repressor, suggesting to us that arginine is present commonly outside, so that you need often to transport it, and rarely to produce it from the inside. And by the way, those two regulators are the same. Arg repressor acts as a repressor for the biosynthesis genes and as an activator for the transport genes. By just shifted sites. All right. So the evidence is around, let's say, 100 different gene systems. Yes.
Don't you usually have more than one level of regulation for the same pathway? Like even the lactoperon has an activator and a repressor.
Good point. So many genes, most genes have more than one, right? So, and we'll talk about that after the break, and what happens when you have multiple genes. So first comment is, if you have two regulators, there's 4 possibilities. Activator, repressor, repressor. 4. If you have n, there's 2 to the power n. Are the rules for those 2 to the power n? You can say that starts becoming pretty amazing, right? To choose between all these possibilities. So let me refine. So each regulator usually has its own signal. So let's call the signal low demand or high demand. And then we'll talk about the 2 signals for the 2 transcription factors separately.
If we're trying to say that activators and repressors act in the same way, that they're symmetric, you could say that the difference between them, the result of the demand rules, is because when we're at saturation, then we're less sensitive to the slight differences in the signal. So if we want to be mainly activated, we'd rather be with a lot of activators and almost in saturation, so we're robust, and vice versa when we want to be repressed most of the time.
So you're saying that activator concentration and activity of the gene, you want to be up here so that variations in the activator have small effect, whereas the repressor you want to be here in this area. Something like that.
No, when
There.
For the graph on the right, if we're just talking about activators, if we want to be activated most of the time, we want to be in the saturation area. For things that are in low demand, we want the small variations to make, if we would have had it as repressors, then the small variations would have made a bigger difference.
Okay. It's nice argument. I think when we analyze the system, we couldn't get an effect out of this particular effect. We tried it, but of course we could have been wrong. So I'm not going to get into it. It's just to say good thinking, and maybe if you have some energy to try to see where this goes. And I wrote the reference here, if you're interested in this, to explore it. Probably you don't have time for it, but yeah.
Aside from the evidence, there's no theory to justify it?
Yes, there is theory, and that's what I'm going to talk to you about, the theory. Yeah, thank you. So let's get on with it. So Savageau had a theory, but this theory is valid. It turns out to be valid in very, very unusual circumstances, and there's a much more robust theory that I'll tell you. And I'll tell you first of all Savageau's theory about mutations. We can learn from it. And then talk about the theory that has to do with specificity. Okay, so okay. So Savageau had made an argument that has to do with kind of a mutational argument. He said something like this. Suppose you have a high demand gene with an activator. Now, if there's a mutation in the activator, most mutations cause loss of function. You lose it. You lose it, lose the activator. As a result, the gene is always off because there's no activator. But it's a high demand genes. That means a great reduction in fitness. And therefore mutation is purified away. So the activator in high demand gene is kind of stable. Any mutation in it, you lose the gene expression, but since you need that gene so often, that causes a high fitness disadvantage, and those mutations are lost. And the activator is, you can say, stable against mutations. In the same situation, if the same high demand gene was regulated with a repressor, mutation would mean loss of the repressor, mutation in the repressor, which leads to gene always on. Because that's definitely a difference between activator and repressor, right? If you get rid of the activator, gene is always off. If you get rid of the repressor, gene is on, right? But that's okay, since we're in high demand. Okay, so it's on, usually it needs to be on. Therefore, less of a fitness reduction. Of course, the mutant doesn't have as high fitness as the wild type because you still need that repressor. I mean, sometimes it is off. Sometimes it is supposed to be off. But it's a less of a fitness disadvantage. Therefore, this mutation has a chance to take over the population, and therefore the R mode is lost less often, right, than in the A mode, the activator. So that's kind of a stability argument. Did I explain myself? I managed to explain this complicated idea. It's not so easy to understand, but what's the problem with this idea? And of course, the opposite for the repressor. What's the problem with this idea? Or maybe there's no problem. Maybe it's a nice idea. It's kind of nice, no? It builds on definitely a definite difference between the two systems. So that's.
Or did you want to say R modes those more often, and therefore you end up with the...
I do. Thank you. So in the end, you see a mode for high demand genes as a result of this difference in stability against mutations. Okay. So that actually works, but it works very rarely, because well, any questions about this explanation? I mean, how? The problem, if there is an intrinsic fitness difference between the A and R modes, it will take over. It will be more important. This mutational arguments are, you might call them second order, because they depend on both cases having exactly the same fitness, so that this, or if I want to say, if I mix together two species, one with A, one with R, for this to work, they have to have exactly the same fitness, so that these mutational effects, where you have a mutant that's less fit but somehow takes over the population, can ever be seen. Because if you mix together two designs, and one has a fitness advantage over the other without mutations, it'll take over just like that. And these mutant effects will have no chance to be seen. Because you need to have this selection of a mutation that's less good than wild type, which means like throwing a coin and getting heads, 100 times in a row or something like that. Because it has to be selected even though it's less fit. So this kind of arguments don't work except in very, very special situations. And there's some papers showing you need that the difference in fitness between the two modes will be less than something like 10 to the minus 7, which is unrealistic. But so let's look at a different explanation. Something that builds on the intrinsic differences between the two modes. Can I erase already? No? Okay. Thanks for saying. If you don't understand this, don't worry about it. It's very intricate, and you can think about it later if you watch the videos. What I'm going to tell you now is much simpler. So don't worry. Can I erase? Okay? Yeah.
Can you maybe explain what you mean by intrinsic fitness difference?
Yeah, I'm going to do it right now. It's something that's built, you can say that it's really inherent in the difference between activator and repressor, and where if you have high demand and low demand, it affects fitness. We're going to take a look at the same problem from the point of view of specificity. So we're going to say that the promoter is inside a cell that has about 200 other regulators, right, who can bind it non-specifically. So The problem is that regulators, they bind to DNA not only to their site, but in other sites. That's a problem of specificity. For instance, the lac repressor spends 90% of its time bound to the wrong places. That's typical. Lac repressor, for example, spends 90% of the time bound non-specifically to DNA. So if you have your promoter and you have your transcription factor that wants to bind it, there is X1, X2 to X200 that want to bind the same site with weak affinities. And in each condition, the activity profile of those 200 transcription factors, whether they're in their active state or inactive state, or whether they're in their DNA binding state or not, changes. So you can't even predict what will be the chemical profile of those transcription factors, because it's not only their existence, it's also their activity. That's a problem. And so you can say we want to detect our specific signal and be insulated from the 200 other signals that change with each environment, right? In other words, if I want the lactose system to make 60,000 copies of the lac repressor at a certain level of lactose, I don't want to care what's the pH, what's the osmotic pressure, what's the temperature, whether there's arginine, leucine, histidine. I don't want to care about those things. I want to insulate my input function from those other inputs. I want to be specific. I don't want the other transcription factors in the cell to change the expression level that I worked so hard to optimize to get to the maximum of the fitness function that we saw. I don't want them to do that. So if I have my signal, and this is the promoter activity, right? How much gene expression is going out there. So I want to be here. What am I doing? No, sorry. Erase that from the video. Here is the protein level. And here's fitness. For example, growth rate of E. coli. We saw that you have this let's say our protein here, this makes protein Y. It's called protein Y. We have this optimal level, right? But now, due to the effect of those other transcription factors that can bind the promoter, they can either shift it up or down, and I can't even know what they'll do. If they're activated a little bit or repressed a little bit, they'll move me. This is the effect of the other regulators, which is non-specific. And no matter which way they move me, they decrease my fitness. And I have a fitness decrease because of this non-specificity. So that's what's called the error. And that's what I want to minimize. And the difference between activators and repressors is now going to help us optimally minimize the error. Okay? Maybe I just say the size here, okay? So a 1% change in, let's say, lac level leads to 10 to the minus 4 change in fitness, which is selectable, which means it's more than the 10 to the minus 7, which is the minimal selective advantage. If you have two strains that differ by more than 10 to the minus 7 in their fitness, random effects like mutants taking over, even if they're less fit, are not important. Genetic drift is less important than selection. So even tiny effects are selectable here. All right. So what does this have to do with the demand rules? You know, I'm going to tell you after the break. Yeah.
Does it happen often that you have non-specific binding when you have a very long DNA?
A very long DNA? Why do you ask?
Because I'm assuming that if you have 200 transcription factors, but you have millions of bases, then the chance to bind a specific site is small.
Yeah, so, right, the more DNA you have per repressed transcription factor unit, the less non-specific binding you have. The thing is that if you have more DNA, you also have more cell volume, and the concentration of the transcription factor goes with that. So it's not only the number 200, but how many total copies there are in the cell. So if there's about 1,000 copies of each transcription factor, there will be a few 100,000 copies of transcription factors in the DNA. Each bounds about 10 base pairs. So you multiply that, you get 1 million base pairs, which is basically the size of the DNA. And that means that a lot of the DNA is bound non-specifically by transcription factors, even weakly. And it doesn't have to be very, very strong binding. It's there, then the RNA polymerase can't bind even 1% less, that's enough. Yeah, to answer your question? So also in eukaryotic cells, instead of 1,000 copies like in bacteria, there'll be 10,000 copies. Things scale up with the size of the DNA, typically the volume. Yeah.
Affinity shouldn't be a problem, right?
Say again?
But affinity shouldn't be a problem. More highly affinity, if a protein has more highly affinity to the site, it will bind. It will displace.
Absolutely. So the correct transcription factor has high affinity to the site.
Okay.
But when you make a high affinity to a site, it also tends to love DNA in general.
Not a specific binding site.
Yeah, so it's like lac repressor has high specificity to this lac site, the string. But as I said, if you look where it is in the binding, it's binding 90% of the time to non-specific DNA. Loosely, very loosely, yeah. So exactly, so we'll see how, exactly, so the high specificity of the real transcription factor is going to help us. Yeah, unless it's not bound.
Yeah.
That's the secret. When it's bound, it's fine. When it's not bound, everything's open. And then that's the point here. Imagine there were, like, for each one of you, 200 other people here trying to kind of non-specifically bind to your seats. You go on a break. You go out to the break. What are the chances that your seat will be occupied? Okay? But you come back and you say, "Excuse me, that's my seat." And that person says, "Oh, sorry, sorry," and they go away, and you're sitting there, right? Now those other people are still here. What's the probability that they'll come and pull you out of your seat and sit in your seat? It's very low, right? So when you're sitting in your seat, you're protecting it. But when you go out on a break, your seat is open for non-specific binders. Okay, and that's what I'm talking about here, is when you bind, that's the thing, when you bind your promoter, there's less errors, non-specific errors. Would you agree with me? It's very logical, right? So that's all, all I want to say. So it's as simple as that. As simple as that, I want to say, I want to say that when the cognate, when the regulator binds its site, there is less error, nonspecific binding, than when the regulator is unbound. In other words, when a regulator is unbound, you have all these non-specific binding possibilities. When the regulator is bound, these non-specific bindings less happen because you're sitting in your seat, basically. Okay, that's one piece of biophysics that we can agree on. Therefore, to minimize error, design regulation to bind most of the time. So if you choose you have two mechanisms, choose the one that's bound to the DNA most of the time. Okay? And here's where the difference between activators and repressors. So for high demand gene, okay, activator mode of control, it needs to be, high demand genes needs to be usually on, right? So activator mode of control is bound most of the time. For a low demand gene, which is usually off, the repressor mode of control is bound most of the time. If you make a high demand gene regulated by a regulator, by a repressor, how is that high demand gene turned on? The repressor is off the DNA most of the time. That means the site is exposed most of the time. That means it's open for nonspecific binding. That means that nice expression level that's needed most of the time changes by a few % between different conditions because of the nonspecific transcription factors in a way that you can't predict because there's so many different conditions and combinations of those 200 transcription factors. I mean, maybe you could say, okay, if we're usually at high pH and there's going to be this nonspecific binding from this pH response transcription factor, I'm going to make my site from the beginning a little bit less strong. But you can't do that for all the combinatorials of 200 conditions which you don't know, right? So you can't design it away. So what can you do? You use, like in engineering, a normally on switch or a normally off switch. These are switches you buy. If you know your switch is going to be normally on, you buy it so its failure rate when it's on is low. And when you know it's normally off, you buy it when its failure rate in the off state is low. So if it has, let's say, a spring in it that needs to be stretched, you make sure that the spring is stretched rarely. You can design two kind of faucets, let's say normally on, normally off. So they have a failure rate in the state that's rare. And that's similar to this idea. I hope I explained myself why the idea of sitting in your seat so nobody can sit there means that you want to sit in your seat most of the time during this course. If we made this course mostly recess and 10 minutes of lectures, right? And mostly recess, it would be a different design, maybe more fun. Not for me, because I like so much to lecture to you guys. Okay, so that's the idea. Let's move on. Let's see what we can do with it. Okay, yeah.
The
Yeah.
So this argument works best if the transcription factor are huge and one transcription factor occupies the whole promoter.
Yeah.
So is this the case? What is the footprint of a typical?
Okay. So here now we start to make predictions, right? We want, like Jean is saying, it's good to if we had a big behind and we could sit on a whole row like this. This would be great because we could block. Okay, so the transcription factors bind sites, let's say, of 10 base pairs, but they can do things like that. Like the lac repressor is actually a tetramer, and it binds 10 base pairs here and 10 base pairs which are far away on the DNA, like far away, let's say 100 base pairs away, in a way that makes a loop. And a loop is one way where you can really protect their. Other kind of transcription factors actually sit on the DNA all the time, and the signal just makes them switch between active and inactive conformation. It's another way to keep the site protected. So it could be also in higher organisms. I don't know how this works in higher organisms, but it could be something with the chromatin coverage of the gene that also has roles like this, because chromatin can really protect sites. But I'm not an expert in that, so. But maybe this idea applies there too. You can make a small calculation. Basically, calculation now I'm going to do just a little bit of algebra, but it's not much more than this idea, which is in words, right? You can. I hope if by the way, if I didn't explain myself, it's a time to ask, okay? Because I'm not. So, if we call this this fitness reduction, this is the error, right? The fitness reduction. I want to calculate what's the condition where we go from activator to repressor. What's the selection boundary? And I'm going to do it here. And then we're going to go to multi-inputs and see how you can, this idea can give you some interesting insight. But so we're going to do it like this. We're going to say that let's call the demand, which is the probability that the gene is on. It's the fraction of time that the gene is on. I'm going to assume here a gene with two states, on and off, just for simplicity, okay? Gene with two states, on and off. And P is the number between 0 and 1. This is what fraction of time it's on. That's the demand. P equals 1 is high demand. P equals 0 is low demand. P can be 0.3, 0.7. So it's a continuum, right? Okay. The error, delta F, due to the open promoter state, like the unbound promoter state, is there's delta F on and delta F off. What do I mean by that? I mean, when the gene is on, you have this fitness drop. When the gene is off, you have a different fitness function. And because of the noise, you have a different. So this is the low expression and the high expression. So that's called delta. The error when the gene is on, the non-specific binding error, and when the gene is off, the non-specific binding error. Now in a repressor, the error comes from. So in a repressor, let's think of a repressor. When does the error occur? When the promoter is unbound. That's when the repressor is off. When the repressor is off, the gene is on. So error comes from the on state. So the error of the repressor is the probability to be on times delta F on. That's the error in the repressor. That's because error comes from the on state, which is the open promoter, the unbound promoter. For the activator, the error comes from when the activator is not bound. Activator is not bound means the gene is off. So when is the gene off? It's 1 minus P, that's probability that it's off, times the error that's associated with the off state. with the non-specific binding to the off state. Okay? So we have two errors, and we can ask when does when does activator win over the repressor? So the repressor has lower error, wins when ER is less than EA, which means that P delta F on is less than 1 minus P delta F off, which means that P is less than 1 over 1 plus. Okay, so what I do, I move this P delta F on here, so if I have P delta F on plus delta F off is less than delta F off. So I think it comes out to be P has to be less than something, right? Delta F on divided by delta F off. So basically it means that when P is small, okay? See, when P is small, that's to say demand is low, that's where the repressor lives. That's where the repressor is better. P is smaller than something. What that something is depends on the gene, depends on what the fitness function looks like, the curvature there, at the on state, the off state, that can depend on different genes. So each gene will have its own transition point, you can say, or where demand. So this is just to quantitate when you have a repressor and when you would have an activator. So just this. Okay. Questions?
Why is delta F on different than delta F off?
Because in general, what's this delta F? It's you think of that fitness function we described where you look at protein level in a certain condition and the growth rate or the fitness, and there's a maximum, and you want to be right at the maximum. And the non-specific error makes you go off the maximum. That causes a reduction. But the shape of that mountain determines basically how much fitness you do. So it could be that in the on state the mountain has this shape, and the off state it has a different shape, and so the errors could be different. But let's imagine that they're the same. If the same errors, this is 1/2, right? This is one. So you can say that if the gene is needed less than 50% of the time, repressor, more than 50% of the time, activator, that's like the demand rules. But in reality, it'll be gene specific a little bit. But if P is close to 0, you can all be guaranteed to be a repressor according to this argument. If it's close to 1, high demand, guaranteed to be an activator. Okay, that's just a little bit of algebra to put this into equations. But so this explanation is in this paper here if you're interested in PNA's 2006. Erez Deckel again and again. Great idea. Tzvik Lusti that we talked about yesterday about conformational, not yesterday, last course about conformational proofreading contributed to this idea too. Now let's think about two inputs. Can I erase now? So it becomes kind of cool to think about this idea when you have 2 or 2 to the power n inputs, because you can make some interesting predictions, suddenly understand some things. So just a canonical example of genes. Usually, actually most genes have 2 or more regulators. For example, the lac promoter that we've been talking about all the time, this LAC-Z, has 2 inputs, a repressor and an activator, actually. Two inputs. So this has activator and repressor mode of control. What are the signals? They have two different signals. Lac I is active, binds DNA, I mean, when there is no lactose. And CRP is active, binds DNA, when there is no glucose. So what's going on here is a system that needs to measure. It's responsible for eating the sugar or lactose, but E. coli has preferred sugar, glucose. And when glucose is around, it shuts off all the other sugar systems. This is something really great about E. coli sugar systems. When glucose is around, it's so delicious, E. coli shuts off all sugar systems, all other sugar systems, all other, like lactose, by basically CRP falling off the DNA, or CRP being inactive. Are you checking me? Am I right here with the logic? Okay. So, so actually, I also want to tell you another thing, that glucose and lactose are both rare in E. coli environment. Why is that? Because our body also loves the glucose, and we have great glucose pumps, so the glucose in our food gets pumped up by our body and other bacteria so quickly that E. coli doesn't see it so often. And when it does see it, it makes sure to eat it very quickly. And so it's rare. You can imagine the world of the bacteria, like fighting over these, squabbling over these sugars. It sounds. Okay. So why AR mode of control, not AA, RR? So we can ask, why AR, not AA or RR? Okay, we can ask. And we can answer from the demand rules. Okay, so let's analyze the situation. There exist genes with AA, genes with RR. They exist, right? It's not that they don't exist. So let's analyze it. Okay, so we have a glucose, lactose, expression. How much of the gene is made? Yeah. So these are the two signals. So we can have low, 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 high, high, low, high, high. These are, let's say there's 4 environmental states. L is low, H is high. And if you look at the gene expression that's needed in each of these states, you see the following. When does E. coli make the most of the highest lac? When there's no glucose and yes lactose. That's when it makes. That's when it's designed to make the most. Okay, so this we'll call expression level 1. It's the highest. When does it make the lowest expression level? It's most repressed when there's glucose and no lactose. That's expression level 4. That's the lowest. Does that make sense that 1 is the highest? No? Should I make it different? Okay. I'm going to drive you crazy. So this 4 and this 1. So 4 is highest expression level. I wanted to rank them. Like 4 is the highest expression level, right? Plus, 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 plus. There's this plus. What about the intermediate states? So it turns out that there are 2 intermediate states: low glucose, low lactose. This will be level 3. Level 2 plus plus, and this will be level 3 plus plus plus. Okay, so this is the highest level. And I'm going to just tell you that this doesn't have to do with regulation right now. You can get this, as I said, with any regulation: activator, activator, repressor, repressor. You can get this with this mapping between outside and inside. You can get it with any regulatory scheme. This comes from the biology. So here you eat the lactose because there's no glucose, there's lactose. Here, this is a funny case, but when there's high glucose and no lactose, you just you shut off the system completely because there's glucose, and you eat the glucose, and there's no lactose. So this is, you shut off the lactose system. When there's neither, you make a little bit of the lac proteins. You prepare for possible lactose, because what you do is you make a little bit of these pumps. So if the lactose comes, you can pump it in. It can bind the lac repressor and turn on the system. You have to have those pumps at a low level in order to detect the sugar. So that's the prepare. And this situation, we have both glucose and lactose. There's other mechanisms that actually shut you down by shutting off the pumps. So this is a state that's you might say I want to induce the lactose system in order to eat the lactose together with the glucose. But in fact, E. coli doesn't want to do that, so it shuts off exclusion by shutting off the pump. That's a detail. It's not so important. Now let's think about 3 different activator, activator, repressor, repressor, and repressor, activator. These 3 possible modes. And consider which of these 4 expression states now is the most error prone, and which of these states is the least error prone. Okay? So activator, activator, here's how the logic goes. Okay? So the activator, activator, let's look at the highest expression level. This is the highest expression level. Activator, activator mode, the highest expression level means that both activators are bound, right? So here, it's bound, bound, and that's the least error prone state. But in the repressor mode, in order to get high expression, both repressors, highest expression, both repressors are unbound. Unbound, unbound. B is bound, U is unbound. All right? So this is now the most error-prone state. So you see, they really, these different modes, you put your error-prone state in different places. Okay. Let's fill it out. So activator, activator, what's the most error-prone state? It's the one where they're both unbound. That's the lowest expression, okay? Unbound, unbound. That's the error-prone state. This is the error-prone state here. And then you have the bound, unbound. So when one of them binds, bound, unbound, and unbound, bound, then you get the intermediate states when one or the other binds. Right? It depends which of the 2 activators is stronger, you can say. What about repressor, repressor? The shut-off state is the one that's most, it's the best. Okay? And then you have the bound, unbound, and unbound, bound. And what about repressor, activator? So repressor activator, it depends which one is actually more dominant, you can say. So let's say dominant means which one has a bigger effect on transcription. So let's talk about the repressor dominant. So repressor dominant means that it affects transcription more than the repressor. So let's first think. What's the so you'll see that for instance the repressor dominant, this is the this will be the bound state. This will be okay. So when they're both unbound, you'll get a different level. So each one of these has a different like mixing. So for instance, you have this one will be unbound, unbound, and then you'll have unbound, bound, and bound, unbound. So the best state will be here. No error. So the point is that the most bound state, most protected state, is different in each of these mechanisms. And out of the 4 possible states, each mechanism kind of prefers a different state. So the question now is in the environment. So according to what's our principle? Our principle is make the promoter bound most of the time. That's our principle. So if we now look at the environment, what happens most of the time? So most of the time, no glucose, no lactose, right? So most of the time, it's expression level 2. So this system is an example of a system where most of the time, it doesn't need to be fully on or fully off. It should be at level 2 out of 4. 1 next to lowest. That's the way it is. Therefore, you want this one to be bound most of the time. And what design does that? Make a RA design. Just like it's observed, right? As observed. With flag I is the repressor and CRP is the activator. And you can also figure out, based on the relative frequencies, whether the repressor will be more dominant, have a larger effect than the activator, everything. Yeah.
It might be not just the question of where it's supposed to be most of the time, but where the price of the error is the highest. You see here where there's low and low, where the cell doesn't have enough carbon, doesn't have what it needs. You want 0 error because a small difference could kill the cell. And when you have lots of sugar of both kinds, you don't mind having a lot of error because you have whatever you need anyway.
Good point. So into this, that's why I did that formula with the delta F. So what I'm doing now is a verbal argument that basically you can say assumes that the error implications in each state are the same. But if you really need to quantitatively add, if in one of the states it could be rare but has such a huge error that it could actually affect the conclusion. That's correct. Yeah. So that's a good point. Yeah.
Would you expect that actually, following the theory that almost all sugars should be regulated by repressors, but there can be only, in some sense, there can be only one sugar that is there more than 50% or more of the time. So this will be the one that is regulated by an activator. and all other sugars should then be regulated by repressors or pressed. But there should be much more sugars regulated by repressors than by activators. Well,
so yeah, the question is, if we say that repressors are for demand of 50% or below, then since there are so many sugars, it's unlikely that many of them will be in demand more than 50% of the time. So most sugars should have repressors. It's a good. I have to think about that. I'm guessing that because of this this two-input situation, it could be that what happens is that presence or absence of glucose is very rare already. But then when there's no glucose, I don't know. I have to think about that. Because in fact, there are many sugars there are with the activator design, like arabinose. And when does that happen? When does activator design happen? We see that the preferred state here is the eat state. What do you mean preferred state? I mean, that's predicted to be the most common state in the environment. What does that mean? Then in the environment, you have no glucose and yes, this arabinose. So the most common state is high expression. And then you want to make that the bound state. And then you look at the table and say, okay, activator. You guys can imagine what happens if you have a gene with 3 inputs or a developmental gene with 20 inputs. And you get big tables like that. And the more inputs you have, the more grades of expression you can have. With 2 inputs, you can have 4 grades of expression. With 3 inputs, you have 8 to the power 3, 8 grades of expression. And then you have all these different 8 possible designs. And depending on which of these states is the most common in the environment, you have an optimal design from that point of view. See what I mean? So you can now observe these systems and guess what's the most common expression level. Is it highest, second highest, third highest, fourth highest, seventh from last, whatever, and maybe have hypotheses about, very fine-tuned hypotheses about the environment.
Were there any evolution experiments that have been done in this, like recording high glucose and high lactose and see if the regulation changes?
So this question is about whether you can experimentally test a theory like that by taking a system with an activator and growing it, let's say, in a low-demand situation, see if it changes to a repressor. So no, I don't know of any experiments like that. But that's the way to think. Of course, there are examples that evolution did this experiment, so that you have systems like I think it's the mating system in yeast, where if you go from strains that have mating very frequently to strains that mate so rarely that scientists didn't even know they had mating till a few years ago, and you look at the regulators, you see the same regulator, but with a mutation that turns it from an activator to a repressor, according to this principle. So evolution could maybe make this experiment something to track down. But one set of experiments that was done recently by Shachrai Malsel, 2011, maybe 12, is to test this insulation idea. So she took a promoter and this regulated by CRP, but designed it so the CRP is one time is a repressor, another time is an activator. And you get the same expression level, but now move that strain into 12 different conditions and ask, doesn't have anything to do with glucose, pH, temperatures, and ask how variable that expression level is to changes in conditions, basically about the insulation. And you could see that in conditions where there was bound, like the high expression for the activator or high expression for the CRP as an activator, or low expression for CRP as a repressor, those had higher insulation, less variability between environments. So there's evidence that insulation exists, but not this evolutionary argument in the lab yet, which is probably not easy, but feasible to do. I just want to tell you that this kind of thinking is not limited just to genes and transcription factors. You can think about it about any level in biology where things need to bind. or to be chemically modified.
Uri?
Yeah?
Very good. There's you have two columns that say RA. The third one you want to say AR, I think. And the table.
Here?
Yeah, this should be RA, and the one to the left should be the other way around.
This should be AR,
Yeah.
and this should be RA.
And you can still.
Okay, thanks. Thanks. This kind of argument, in principle, could work for any piece of biology where you need to save your seat, basically, just like we discussed here. For example, if you have a protein that's phosphorylated, and its same site could be phosphorylated or non-phosphorylated, this could be the active state, and this could be the non-active state. Or you can design it so this is the non-active state, and this is the active state. So what I'm saying is that you can have a protein and design it to be phosphoactive or phosphoinactive. And there's examples of either, where phosphorylation activates a protein or phosphorylation inactivates a protein. Different enzymes. And now you can say, are there rules to determine whether a protein is phosphoactive or phosphoinactive? Are there rules, right? And based on what we said here, you can imagine that the question is, which state is more error prone? Okay, so if the phosphorylated state is more error prone, that's to say the phosphatases make mistakes more than the kinases, the ones that put on the phosphate. If this is more error prone, okay, then high demand function would be phosphoinactive. Why? Because if it's a high demand function, you want it to be less error prone state most of the time. Therefore, you make this one active and this one inactive, for example, right? But if kinases, if this state is more error prone, which means the kinases make mistakes, so if you have this state, some non-specific kinase can phosphorylate it more often than if you're phosphorylated. Some non-specific phosphatase can take off the phosphate. Then you have the opposite. High demand means phosphoactive. See, so you can, with this principle, you can, in principle, apply it to other modes of this kind of specificity, or all kind of binding things, whether things bind to activate you or bind to inhibit you. This happens, there's, in every biological system you have examples of this, of binding or phosphorylation, something activating and inactivating. But now you can ask, why this particular design? What does it tell us about the frequency at which that function is needed in the normal conditions of that cell type or organism? And I think it's pretty much open. For example, this is open question for research. All right. So to summarize, we saw here how this idea of keep your promoter bound in order to reduce non-specific binding predicts that, this demand rules, how high demand activator, low demand repressors, and gives us, more importantly, I'll just end by quoting Michael Savageau, who deserves a lot of credit for this way of thinking. This is how he wrote. Okay, he said in 1989, biological mechanisms are rich in variety, and different systems often have different biochemical details. How can we understand this variety? Differences in biochemical details might be the result of historical accidents that are functionally neutral, or they might be governed by additional rules that have yet to be discovered. One can always assume that certain differences are the result of historical accident. But such an explanation has no predictive power and tends to stifle the search for alternative hypotheses. It generally tends to be more productive if one starts with the working hypothesis that there are rules. One may end up attributing differences to historical accident, but in my opinion, it is a mistake to start there.